Thanks. It's great to see everybody. Happy New Year. Cheers to 2022. Hopefully the road gets a little bit smoother as we peak the crest of Omicron and we can start meeting in person again. That's my hope. But thank you all for tuning in for our Science Talk webinar series. We're kind of at the tail end of our Research Thrust B series, which focuses on the impacts of innovative technological solutions for urban water systems. So we do have one more webinar in our series that will be next week, and we'll hear from Arpad Horvath over in UC Berkeley. He's going to talk a little bit about life cycle assessment of urban water systems, but today we're going to hear a little bit more about greywater reuse and pathogen removal. As y'all know, we are recording these and I post them to our YouTube channel as well as the UN website, but feel free to use the chat queue to enter your questions or turn on your mic and webcam so we can have a bit of an engaging discussion as we wrap up the presentation. So with that, I am excited to introduce our presenter for today, Dr. Kimberly L. Jones, Professor of Environmental Engineering, Department Chair of Civil and Environmental Engineering, and Associate Dean for Research and Graduate Education at Howard University. She holds a BS in Civil Engineering from Howard and an MS in Civil and Environmental Engineering from the University of Illinois and a Ph.D. in Environmental Engineering from Johns Hopkins. Dr. Jones's research interests include developing membrane processes for environmental applications, physical chemical processes for water and wastewater treatment, remediation of emerging contaminants, global drinking water quality, environmental justice, and environmental nanotechnology. Sorry, I stumbled through that a little bit. I guess I'm not properly caffeinated, but I'm ready to turn it back over to you, Dr. Jones. I'll give you the platform and let you take it away.
Thank you so much, Sarah, for the introduction and for having me here today. I just want to announce I just also took on a new role at Howard as Associate Provost for Faculty Affairs, which is why my week has been so crazy this first week of the semester. But yeah, that's brand new. I don't even know if my students knew. I know Theo and Hamid are up here. Two of my students are on the call. And this is Theodore's thesis, his dissertation project. But the title is Greywater Reuse, Pathogen Removal by a Membrane Bioreactor. So I will be updating you on what's going on, but we'll maybe refer our questions to Theo since he's on a call and I have to jump off soon after this. So hopefully he's ready. I'm giving him a heads up. So just in general, what is water reuse? So when you think about water reuse, there are a lot of different definitions. Some call it water recycling, water reclamation. But in general, water reuse is when water is reclaimed from any of a variety of sources. So the sources could be wastewater, industrial sources, stormwater, and then that water is treated and is reused for some beneficial purpose. And the beneficial purpose can also be very, very broad. Irrigation for agriculture, landscaping. It can be used to augment municipal water supplies in some cases. It's used often as processed water in power plants and other industries. You can use it to flush toilets in your house. So there are a lot of uses for this reclaimed water. And a lot of states are coming up with regulations, and I'll talk about this a little bit later, but EPA does not have hard and fast regulations because the key is, for water reuse, what you have to be mindful is that it's fit for use. So you have to figure out what the intended use is, and then the water is fit for that intended use. And so what we chose to focus on in our project is agriculture or irrigation. I mentioned that as one of the uses. And when we think about sustainability, when we think about increasing or wasting less water, for lack of a better term, we want to think about reusing some of the water reuse that is most impactful. And so here on the left, this is just water withdrawals over the last, from 1950 to about 2015. This study's done every 5 years. So it's probably a 2020 version. But the point here is that if you look at this blue line at the top, this is withdrawals per year or every 5 years. You can see here that the withdrawals was increasing, and then we got a little bit better at how we manage water withdrawal. And then it began to come down, and this decrease, if you look at what makes up these water withdrawals, the decrease is due largely to decreases in thermoelectric power. If you look at the other large use, it is agriculture or irrigation. Part of that is our population is growing. As our population grows, you have to feed more people. To feed more people, you have to grow more crops, you have to water those crops. And so irrigation tends to be a pretty significant part of water withdrawals. And just what most people think about, the public supply, that's this kind of purple supply down here. You can see it's a much smaller percentage, but you should know that that number is relatively stable. And so the biggest decrease is really due to our progress in reducing the thermoelectric power withdrawals. And this on the right, this bar graph shows, you see total withdrawals from 2014, then 2025, and then 2040. So these are projections. And you can see here that the percentage due to consumption is small, but projected to increase a little bit as we go from 2014 to 2040. And this lighter is the total withdrawal. And so that is including other withdrawals. And that number also is projected to increase from 2014 to 2040. So we want to be mindful, given that irrigation is a large part. We thought that coming up with a nice solution for treating water or reusing water that would otherwise be wasted for irrigation was a nice thing to focus on. We're in Washington, D.C., and so we thought urban agriculture in particular was a nice thing to focus on because we're beginning to see more urban gardens. Urban agriculture is a quickly growing industry. We have food deserts in a lot of urban areas. And so urban agriculture is touted as a solution to that because it's a way to educate members of a community about being more green, healthy food choices. So there are lots of reasons why we're seeing more of these urban gardens. So many benefits, but amongst the challenges are water management. Really, a lot of those just use treated water, water that you could drink. They just use it to water these urban gardens. And so water management is something that can be a challenge because we do want to be more sustainable. Potential for contamination by urban pollutants in a large city. You could have pollution to seep in through the air or through other sources. And then guidelines to estimate water use. So what the impact is on our water system. So what we decided to focus on is membrane bioreactors to produce fit for use water for water reuse, particularly with a focus on these urban gardens. And here is just a cartoon to show you what a membrane bioreactor often looks like. The membrane is a size exclusion technology, and it really uses this picture on the right just as a very easy cartoon to understand what a membrane does. You have the membrane here, you have your effluent or your suspended microbes in your bioreactor, and the membrane just filters them. So think of it like a conventional activated sludge filter or conventional activated sludge process where the membrane is the filter. And a membrane is a much more efficient filter than some of the filters that are typically used in a conventional activated sludge system. And so given that the membranes can be fit into a smaller footprint often, and you get more efficient removal of a lot of contaminants that may otherwise go through your filters. When you think about decentralized water reuse systems, when we think about these urban gardens, which was our focus, we wanted to think about a water reuse system that could be set up at the urban garden site. A lot of them have these large rainwater collection systems, and so they harvest rainwater. They don't always treat it. In other cases, they just pipe it in and use it. So what we wanted to do was a number of our urban garden partners stated, some concerns about, they try to use rainwater. Rainwater varies. They ultimately often have to augment it with potable water. And so we thought if we have a decentralized water reuse system that would allow them to use maybe gray water or some other water that would otherwise go to waste, it would be a nice benefit for them. And so here are just some benefits of membrane bioreactors. Membranes have about a 30 to 50% smaller footprint than conventional treatment technologies, biological treatment technologies, which tend to be very large. You can membranes, because they're small, are more easily adaptable. So you can change your operating conditions if you need to, if you have targeted removal. And in our case, what the research need is, and I'll talk about this a little bit later, is figuring out how well membranes remove viruses and bacteria, because that is one thing that is a concern. If you're in these urban gardens, you don't want to increase exposure to these pathogens. You want to decrease exposure. You also don't want to be irrigating your gardens with water that's full of bacteria and viruses. Also, membrane fouling is a priority. It's an issue with all membrane systems. It's something that we are always mindful of. And so reducing membrane fouling is always top of mind, because if you can reduce fouling, you can really extend the useful life of the membrane and increase the efficiency of the membrane process. So we identified some general research opportunities here. We thought that if we can come up with modular membrane bioreactors, we need them to be cost effective, save energy, use, again, think about modular small systems. They can be used locally in these urban installations, and they're more sustainable than trying to depend on rainwater and having to augment with potable water, which is often done. Some of the obstacles are really figuring out how safe the treated water is, as I mentioned, and I'll show later. There's some inconsistencies with regards to pathogen removal by some of our membrane bioreactors, and also educating the folks who are running these sites on the correct use of the membrane bioreactors. And so we thought there were a number of research opportunities for us to think about microbial contamination, how to reduce it, think about ways to increase the use, particularly in urban agricultural applications that may not have a large footprint to put in a large treatment system, could be done in a much smaller footprint. And also to come up in general with more decentralized treatment options, which we could be implemented for a lot of other applications. So just again, just to talk about some advantages of membrane bioreactors over conventional treatment, their increased efficiency at removing pathogens, particularly bacterial, get 5 to 7 log removal of many of your types of bacteria. Also, I mentioned the footprint. You can often you need fewer steps. A membrane is almost an absolute barrier. And so you don't need as many steps to often you'd have a secondary clarifier, sometimes a filter, a sand filter. In this case, you would not necessarily need your secondary clarifier, which tends to be very large and very space needy. But in this case, our membranes are much smaller, which is really why you get that smaller footprint. And also, if we can optimize bacterial and viral reduction, then you also won't need disinfection. Often you have to chlorinate and then dechlorinate, depending on what the use is. But in this case, we could get downstream disinfection by the membranes themselves to reduce the need for further disinfection. And so just real quickly, I'm just gonna kind of show you, I know this is a table with a lot of information, but the main point here is we look at many different types. This table has different types of bacteria. You have different pore sizes for membranes. If it says 0.4, if there's a number here, these are microfiltration membranes. If it has kilodaltons, those are ultrafiltration membranes. So we tend to use microfiltration and ultrafiltration membranes for membrane bioreactors. You can use different configurations here, different materials. But the point is, when you look at the log removal for these membranes, it tends to be for bacterial removal, about 5 to 6 log removal pretty consistently for a lot of these membranes. But when you look at viruses, you can see here, again, you have different types of viruses, different pore size membranes, and most of these happen to be microfiltration membranes. You see here that the log removal is much lower. It tends to be, there's some exceptions here with some of our larger viruses, you may get 5 log removal, but typically they tend to be around 1 or 2 log removal. And so the removal efficiency for viruses is considerably less than it is for bacteria, mainly because viruses are smaller and are able to go through the membrane pores. And so we wanted to really focus on that to see if we could increase the removal for viruses. And here's just another study. On the left is a graphic. I think that Theo made just to show you really what is happening. And it's just a very simple size exclusion situation where the bacteria tend to be larger. They won't go through the membrane. The viruses are smaller. So you have to think of different. This graph on the right really just, again, shows you when you have bacteria and viruses. You see here the log reduction and the ratio of diameter to the pore size of the membrane. And it shows you that for our the smaller material, the log removal is lower. And so we think about specifically virus removal mechanisms in membrane bioreactors specifically. You can see there are three main mechanisms that are typically identified. One is size exclusion. Not only do you have the membrane, but this cartoon on the right attempts to show you this. You have a cake layer that builds up on the membrane surface over time. That cake layer reduces the effective pore size of the membrane. And so by having a smaller effective pore size, you may be able to reject some of those viruses as this cake layer builds up. Also, you can just have decay due to, you have a lot of microorganisms in this feed solution over here. And so you may have some reduction due to predation or other enzymatic activity. So you may get some reduction there. And then biomass absorption tends to be kind of a critical contributor also, but that may be just absorption of the viruses to some of the suspended biomass. So you have a number of different mechanisms that can impact virus removal here. But one thing is it is often difficult to control all of these mechanisms because they're due in large part to live pathogens that are all working in your feed water, in your suspension. And so what we wanted to do is really focus on ways that we could improve it. And just also to look at biomass absorption, you can see here that it's been shown to be can be a considerable mechanism for removal. So what we decided to think about was maybe coming up with a nanoparticle functionalized membrane bioreactor for enhanced pathogen removal and reduced fouling. And so this picture on the left, these are some of our prior just results from some of our work in our lab that we've done over the years. In our lab, we've done a lot of work in general on reducing biofouling of membranes. And so for those projects, what we did was we put a nanoparticle on the membrane surface to try to decrease the buildup of bacteria on the surface, so you reduce biological fouling of membranes. And so we showed that that was possible. We did a couple different things. This picture on the left, these are pictures where we looked at, we did a UV modification of our membrane, and this just shows the percentage of damaged cells. We could control that by increasing the UV exposure to the membrane surface. And then if you look on the right, these membranes, this is a system where we put silver nanoparticles right on the surface of our membrane. So we embedded our silver nanoparticles in a polyelectrolyte. And then we put those on top of a microfiltration membrane. So this picture at the top shows an unmodified microfiltration membrane. And you can see here that we saw bacterial growth on that membrane. This picture at the bottom shows us our modified membrane, and we saw reduced bacterial growth. The issue here is that our prior studies mainly looked at bacteria. That was important. Second thing is we did not look at this in the context of a biological treatment process. And so we have to be careful because we have to think about whether these processes we want to reduce or improve the reduction of bacteria and viruses across the membrane surface, but we do not want these particles, for example, here, the silver nanoparticles, floating around in our suspension because it would impact our biological processes that we need to happen in order for our biological treatment step. And so those are some things that we're thinking about. We've outlined a couple of research questions here when we think about our modified membranes. can we get enhanced viral inactivation, and how would we do that? So we need to think about the impact, and there's been very little work done on what the impact is on viruses. And we may need to look at different types of nanoparticles. Again, we looked at silver, which is just is a known has exhibited known cytotoxicity. So we knew that silver is likely to reduce bacterial growth, but we didn't really look at viruses and how the reduction might be. And also thinking about the structural properties of our membrane, and think about what the processes are when we think about oxidizing the nanomaterials, or what really is going on the surface to enable this reduction. And so here I just outlined just general treatment goals for irrigation. These are goals, they're not hard and fast regulations, but these are some target values. And you can see here, one thing I want to point out, there are a lot of things that are mentioned. You don't see necessarily viral reduction. You see for irrigation, they mainly talk about coliform reduction, but we also want to include as a target what some log reductions may be specifically for viruses. I want to mention really quickly, we have been trying to work with an industrial partner to try to increase technology transfer, because we, for an application like this, we want to make sure our final process kind of gets out in the hands of folks who could use it. So we were trying to pursue a partnership with Suez and also some of our urban garden folks. The pandemic has really inhibited this partnership to some extent, because they've run into supply chain issues. We haven't really been able to get the membrane bioreactor system on campus. So we are pivoting to possibly another source for our membrane bioreactor, but we're kind of trying to figure that out at this point. But I think in these talks, we're asked to talk about community benefits, or benefits to certain communities. And so if we can, and I mentioned some of this before, but we're really hoping that we can come up with a system that is easily retrofit into one of the commercially available membrane bioreactors, where we can come up with either a dosing mechanism or some way to get these nanoparticles onto the surface of the membrane that's relatively inexpensive, so they can be shipped out and used in some of our urban gardens. So we're thinking about a modular system to be used to treat greywater or stormwater for urban agricultural applications. We also wanted to come up with educational modules for outreach to the community, to really also use this as a way to educate folks about water reuse in general, and ways that they could come up with what for them may be unexpected ways to reuse water. And also, if we can come up with ways to make these urban gardens more sustainable, we may increase these options for healthy food choices, particularly in some of our communities with food deserts. And so just to give you an idea of the scale or who might benefit from this, we have here, Washington, D.C. on your left. This is just a map, and this odd-shaped thing is Washington, D.C., right here between Maryland and Virginia. And so just to show you, we have a number of urban and community garden installations in the works in the DC area. Over here to the right is a zoomed in picture of these. We have a collaboration between the DC government and community organizations to really increase the number of urban gardens in DC. What they're trying to do is reduce the food deserts and sometimes fresh produce deserts around the city, come up with more ways for folks to grow their own food, maybe have farmers markets. Farmers markets tend to be in more affluent neighborhoods. We want to have more farmers markets all over the city, in communities where they may not traditionally have access to fresh produce. And so in that way, it's an educational opportunity and a way to promote good physical and mental health. We also hope to increase engagement on our campus. We've been wanting to put our system on the roof of our interdisciplinary research building. We had gotten permission. With the pandemic, we have not been able to get back to our efforts to put these on the rooftop. We are talking about other green infrastructure installations on the rooftop, so we're hoping we can get our membrane bioreactors set up along with the other green infrastructure installations on the rooftop. And that way we could have these systems to bring other undergraduate students into this project, because this would be a very nice way for them to kind of become interested in sustainability. We have a pretty robust sustainability effort on our campus. And so we have students who may not even be in engineering necessarily, but they may be interested in learning more about sustainability, learning more about water reuse. So we're hoping that we can use this to engage members of our campus on water reuse and sustainability. So with that, I would like to thank you. Here's my contact information and contact information for Theodore, my graduate student. And I will stop now and take any questions that you have.
Great, thank you, Kim. Any questions for Kim or Theo?
This is Paula Kehoe. The fantastic presentation. That's very exciting of all the work that you're doing. I just also want to let you know we have a lot of resources on log reduction targets that we've been doing under the National Blue Ribbon Commission for on-site non-potable water systems. It's not specifically related to agriculture, but there is a connection. And so we'd love to share those resources in terms of the water quality health risk-based standards that we've established through this partnership that focuses on protozoa, virus, and bacteria for gray water and black water and rainwater and stormwater. And so just because we'd love to keep following your research and again, opportunity to share the information that we've been working on and collecting over the past several years. And we're also establishing a certification program for operators of small on-site treatment systems that we'd love to share that information with you as well. But that's fantastic work. We really look forward to hearing more and learning more from all of the great work you're doing. So thank you so much.
Thank you. And I'm going to definitely follow up. If you could drop your contact in the chat, then we will absolutely follow up with you on that. Thank you so much for that.
Thank you.
I can connect you too as well.
Okay. Yeah. Thank you.
Thanks for your time, Paula. It's great to see you.
I wish I've had more time to participate in these. These are fantastic presentations. So thank you so much.
Great. Well, if you get really bored in your copious amounts of spare time, they're all posted to the UN website and our YouTube channel. So they're there whenever you're ready.
Thank you.
Any other questions for Kim and the team? All right, well, I'm not seeing anything in the chat and haven't seen anyone else raise their hand or turn their mic on. So Dr. Jones, I know you're a woman in high demand right now and having to scoot off. Congratulations on yet another thing to keep you occupied and running from meeting to meeting.
I know I don't seem to get enough, but thank you. Thanks for having me. This was good. These are great. And actually Theo's been going through reviewing a lot of those. So thank you for posting these on the website. That is really helpful.
Absolutely. Great. Well, it's lovely to see you. I hope everyone keeps their head above water, pun intended there. Take care and stay safe out there. And hopefully we'll see you all next week. Have a great afternoon, everybody.
Thank you very much.
Thank you.